Okay, well, I think we'll go ahead and get started. So welcome, everybody, and welcome to the students from Blake High School who are here joining us today for our Cancer Center seminar. Today, I'm pleased to welcome our speaker, Dr. Pamela Jacobson. She is a distinguished professor and associate department head in the Department of Experimental and Clinical Pharmacology in the College of Pharmacy here at the University of Minnesota. She earned her PharmD degree from University of Nebraska, went on to do a residency at the University of Michigan, where she became a faculty member before coming to the University of Minnesota as a faculty member. And she studies the pharmacology of immunosuppressants and anti-cancer agents, as well as pharmacogenomics, which has to do with how the genes that we inherit influence how we metabolize drugs. And this is important for determining the right type of drug and the right dose of drug for patients. And so this is part of the concept of individualized or precision medicine, and she's the co-principal investigator of the Minnesota Precision Medicine Collaborative, and today she's going to tell us about pharmacogenomics of germline mutations for predicting cancer drug response and toxicity. Thank you, Pamela.
All right, good morning. How's the audio? Everything here? Again, extend my welcome to our high school class here today. I hope I can excite you about an innovation in science, this area of pharmacogenomics. So I'm not going to talk about my own work today. What I'm going to talk about is where we're at in terms of the science of pharmacogenomics and some of the innovations that have been occurring, where it's been rolling out into practice, so sort of give you an idea in terms of where we're at with actually implementing this into care. So this is, I think, as everybody's probably seen, such a picture, but this is the way today's current drug treatment is. That we have all the we have patients with all the same diagnosis, but they all get treated the same, usually with the same set of drugs, same doses, etc. But we have 4 different outcomes that can occur in these individuals. Some individuals have no benefit, and they develop some sort of side effects from their medicines. Some patients will have a positive benefit, but also develop some toxicity. We have some patients that have a positive benefit and no toxicity. Of course, this is where we want everybody to land, that they have a good outcome and don't develop side effects. And we have others who have no benefit and develop no toxicity. So ultimately, pharmacogenomics is targeted to try to get everybody within this green category where we can have positive benefit and no toxicity. So there's a lot of factors that influence variability or response to medications. Certainly dose is very important. We know that from years of study. We know that the ADME properties, which are the absorption, distribution, metabolism, and excretion properties of drugs, are terribly important. Age is important, gender, environment, lifestyle, and diet. We know disease status is important, comorbid conditions, performance status. And now we know that genetic variation that we inherit from our parents is also very important in determining response and toxicity of medications. So this is a pharmacogenomics panel. This happens to be my pharmacogenomics panel. I've been tested twice with 2 different companies. And I have a number of mutations. Most everybody in this audience will have a number of mutations in pharmacogenomic genes. So I'm an ultra-rapid metabolizer for one enzyme called CYP2C19 that has implications for several different drugs. I also carry 2 mutations in CYP2B6, which also has implications for a number of very important drugs. I have 2 mutations in SLCO1B1, which has implications for side effects for statins. So should I ever receive a statin therapy, I'm going to be at risk to develop the muscle toxicities. I also carry one mutation in UGT1A1, STAR28, which would put me at high risk to develop irinotecan-related diarrhea and myelosuppression if I should ever require irinotecan. So even though I appear fairly normal, I am not, because I have a lot of mutations. And we know from many other studies that most people do carry many mutations in the pharmacogenomic genes. So I have 3 little cases I'm going to go over with you just to kick this off. This is a case. This is a pediatric case where they've used pharmacogenomic testing to determine a dose for a child with acute leukemia. So this is a 5-year-old female. She was diagnosed with standard risk Allele. She had pharmacogenomic typing for 6-mercaptan Purine treatment prior to starting therapy. And she turns out that she's an intermediate metabolizer based on her TPMT genotype, and she's a normal metabolizer based on her NUD-T15 metabolism status. So both of these are important enzymes involved in the metabolic pathway of 6-mercaptan Purine, which is a common drug that's used to treat leukemia. Because of that one mutation that she has in her in the TPMT, they decreased the dose by 50% before starting therapy for her. So she continues initial dose reduction. She's currently halfway through her maintenance cycle 3 and has not missed any chemotherapy due to neutropenia and has experienced only 2 occasions where her ANC was too high, which of course there's a balance between the ANC being too low versus too high to keep good therapy. So this is a child that is in care over at Children's Hospital. I collaborate with people over at Children's, so this is one of the cases they provided to me. Here's another case that was reported in the literature just recently. This is an 18-year-old male who has T-cell ALL, who began experiencing unexpected myelosuppression during maintenance phase of therapy. After his first hospitalization, they decided to do genetic testing on this child, and they found that for TPMT, he was a normal metabolizer. So they concluded that genetic that the child did not have genetics that could explain the fact that they were developing fever and neutropenia and having side effects and coming to the hospital. So the child continued to experience neutropenia, resulted in 2 or more hospitalizations for neutropenia. There is now evidence, and pretty substantial evidence, for a new mutation being identified that's important in 6-mercaptan Purine therapy, and that's the NUD-T15 mutation. So they decided to test this child for the NUD-T15 mutation, and it turns out that this child also had a mutation in this particular gene. And this is likely the explanation now for the fact that they were having trouble tolerating their chemotherapy. So this child went on to relapse and then ended up going on to bone marrow transplantation. And they were hypothesizing that potentially the reason this child ended up relapsing and coming to transplant was because they had stopped the therapy so many times because of the fever and neutropenia that they felt that they probably had insufficient therapy. So here's a case where perhaps these mutations may have affected relapse. And this is my final case. This is a colorectal cancer case. This is neoadjuvant therapy. So this is a 64-year-old male that has new onset blood in the stool, occasional abdominal pain and constipation. He presents to his primary care clinic. He has anemia. He's now scheduled for a colonoscopy. He ends up being diagnosed with stage 3 colorectal cancer, and he's going to receive mFOLFOX 6 for treatment. They do, prior to starting therapy, they conduct a genotype test. They look for the DPYD mutations, and it's found that this individual has is an intermediate metabolizer, which provides them an activity score of 1. Many of these different mutations, they have a scoring system where the number of different mutations you have turns into a score, and then based on the score, then they can determine the dose. Because of this set of mutations, it's recommended that this person get a 50% dose reduction of their 5-FU therapy. And that's because that we know that individuals who have these mutations are at much higher risk to develop the myelosuppression, some neurotoxicity, the mucositis, the hand-foot syndrome, which is so common with 5-FU therapy, and diarrhea. And so in this case, now that they dose reduce, what they will do, they'll determine if this patient if the patient tolerates therapy, they might try to escalate the dose very slightly. If the patient doesn't tolerate therapy, they'll continue to dose reduce it. So that would be a typical way that this particular mutation might be used in neoadjuvant therapy for colorectal cancer. So there's many different areas now where pharmacogenomic is being used. We're seeing a lot of use within psychiatry, trying to help is selection of the antidepressants, particularly the SSRIs and the tricyclics. Cutaneous adverse drug reactions is another area. So this is that there's a number of the HLA mutations that are associated with hypersensitivity reactions, Stevens-Johnson syndrome. So it's being conducted prior to the use of many of those drugs. Certainly in cardiology, there's a number of areas where it's being used. For clopidogrel and warfarin are a couple examples. For antivirals, and of course there's now there's evidence within cancer for certain drugs that pharmacogenomic guided medications can be quite valuable. So there's a number of genetic predictors, as we all know in this room, for cancer outcomes. Some genetic mutations are prognostic. Those certainly the somatic mutations have been very, very important in cancer, but many germline mutations also have prognostic predictions. Many of the mutations can predict efficacy, particularly tumor sensitivity. But then the pharmacogenomic or the germline mutations, they're very commonly used and predictive of many of the toxicities. And certainly they're very predictive of pharmacokinetics. And I'm going to show you some examples of where some of these drug metabolizing enzymes and mutations within those drug metabolizing enzymes is very predictive of pharmacokinetics, which of course leads to differences in toxicity. So genetic variants alter two really important key concepts in pharmacology. They can alter the pharmacokinetics of a drug. They can also alter the pharmacodynamics of the drug. In terms of the pharmacokinetics, absorption, distribution, metabolism, and excretion are all known to have genetic determinants. Metabolism, by far the most common and the most powerful form of genetic of association with genetics. Pharmacodynamics, there's also some important, not necessarily in cancer, but in other areas where there's mutations in receptors or ion channels or signaling pathways, the immune system like the HLA and the transporters. We all know that mutations within those can also create pharmacodynamic changes and differences in response. So these would be some typical pharmacokinetic profiles that might be observed in patients with these different types of mutations, of course, phenotype status. So the first pharmacokinetic curve on the top right would be somebody who might be a poor metabolizer. You can see that they have a very high pharmacokinetic profile. The area under the curve would be very large. They have a very high peak concentration. That, in contrast to an ultra-rapid metabolizer, which would be on the bottom would be that this would be somebody who would have very, very high clearance of their drug. And you can see that their area under the curve would be very small and their peak concentration would be dramatically reduced. So there can be extreme differences depending on these mutations and the kind of phenotype that the mutation leads to in the pharmacokinetic profile. So there are a lot of cytochrome P450 enzymes. These are some of those that are involved in drug metabolism. And for most of them, they are known to be polymorphic. So we know that there's a high probability that many of these could result in a change in drug pharmacokinetics or metabolism or through excretion. We know that CYP3A4 and 5, which of course is the main drug metabolizing enzyme for all drug categories across all therapeutic areas, has a number of polymorphisms. CYP2D6 is highly polymorphic. Again, another very, very common enzyme system. CYP2C9 and 2C19. So those would be the four most common ones, and we know they're highly polymorphic and highly influential. So just a moment on the nomenclature of pharmacogenomics. So the nomenclature of pharmacogenomics is a bit different than the nomenclature that's used in traditional genetics or disease genetics. So within pharmacogenomics, they use a star allele classification. So In this example, so it's still all these mutations that are identified for drug metabolizing variants are given an RS number, as all mutations are, but they're also given a star designation. So the example would be here that for this particular gene, CYP3A5, the A to G mutation is referred to as the star 3 variant. Instead of an A to G, it's called star 3. So when you read the pharmacogenomic literature, you see a lot of discussions about star alleles. They all relate back to nucleotide changes, but that does make the literature a little bit more confusing than the traditional genetics. Here's an example. So here's a table that's been created for predicted enzyme activity for 3 of the main pharmacogenomic genes. You can see all these different star alleles that have been assigned. And for many of these, for example, CYP2D6, there's over 100 different star alleles that have been identified, or mutations that have been identified for CYP2D6. So it does make it a little bit more difficult to read this literature. So there's a number of mechanisms by which these mutations can cause alterations in drug metabolism. These would be the primary ways. Certainly, some of the pharmacogenomic genes are completely gene deleted. And a good example of this is CYP2D6, which has gene deletions. Of course, if you have a gene is deleted, you're not going to the patient won't have any enzyme. Of course, then you're not going to have any good. There's very little drug metabolism, so very poor clearance of drugs. You can also have problems within single gene copies, where you can have unstable enzyme that is formed, can have an altered substrate specificity. In the case of unstable enzyme, most likely that there's poor metabolism of the substrate. In the case of mutations that can cause altered substrate specificity, what oftentimes might happen is that other metabolites get formed. So unexpected metabolites might get formed because of that mutation. And another very common way that drug metabolism can be altered due to mutations is through gene duplication. And CYP2D6 is another really good example of gene duplication. So there might be multiple genes now that are inserted within the genome, and in that case, lots of enzyme is going to get produced, and those individuals will have extremely high drug clearance. Another really important aspect of pharmacogenomics is the differences in minor allele frequency across the world populations. This particular slide shows just some of the 4 major drug metabolizing CYPs. And if you just look at the top one, which is CYP3A5, which metabolizes most commonly metabolizes drugs, you can see that the minor allele frequencies is very common across 4 of the 5 world populations. But for individuals of African ancestry, we have very, very different minor allele frequencies. And in fact, they have different types of mutations and variations. And you can see that there's a lot of differences in the colors across all these drug metabolizing enzymes. So the point here is that we do need to do ancestry-specific genetic testing, because there are differences between the world populations. And just because it doesn't occur in the Caucasian population doesn't mean that it doesn't occur in another population. And of course, we know that most of all medications and drugs have been developed in primarily Caucasian populations. So then when these drugs are translated into other ancestry groups and they don't work as well, it probably shouldn't be of any surprise, because we don't understand how maybe some of the mutations that they carry can change the drug itself. So something very important that's happened in pharmacogenomics is the formation of this consortium. So this is the Clinical Pharmacogenomics Implementation Consortium. This is an NIH-funded consortium. They've been writing pharmacogenomic guidelines now for a number of years, and they review the evidence, the pharmacogenomic scientific evidence, and they grade the evidence, and they provide guidelines that are relevant for practice. And so they have been very instrumental in trying to narrow down the drugs where the pharmacogenomic information is strong enough and there's enough scientific evidence that it is of value to translate into clinical care. So this is just a list of some of the drugs. Actually, this is all the drugs, not some of the drugs. This is all the drugs that have been reviewed now by the CPIC consortium. Many of the drugs on this list are highly relevant to cancer. There's cancer drugs. There are a number of supportive care drugs. There's many hematology drugs that are on this list. And all of these drugs now have been graded and have a level of evidence that this group feels that is sufficient for implementation into care. They also they just got renewed, so they just heard from the NIH in the last couple of months that they're renewed now for another 5 years. So we'll at least have another 5 years of guidelines being written and evidence reviewed. There's a number of drugs that they have guidelines that are in progress. So I'm going to go through 4 examples. Only 4 of the examples of the many that we could go through. I'm going to go through an example of CYP2D6, 2C19. I'm going to go through the TPMT or the 6-mercaptan purine example, and then do something with DPYD for 5FU. So this is CYP2D6. This is a messy enzyme. It's complicated. And there's so many mutations within this particular gene that leads to a multitude of different multitude of phenotype categories. So, but there's really three categories we're most concerned about. And these would be individuals that fall in the intermediate metabolism category, poor metabolism category, or the ultra-rapid metabolism category. And those 3 categories are all known now to profoundly affect drugs in one way or another. So the first one I want to talk about is tamoxifen. This has been something that's been under study for many, many years now in terms of the metabolism of tamoxifen. It's known very well that tamoxifen is metabolized by the CYP2D6 enzyme. It's primarily metabolized to N-desmethyl tamoxifen and then endoxifen. And N-desmethyl and endoxifen are highly active against tumor. So anything then that would impair the transformation of tamoxifen into desmethyl or to endoxifen is ultimately going to reduce the activity of tamoxifen, or at least presumably. So this has now been studied in multiple different trials, and there is now a clinical guideline now that recommends recommends using an alternative therapy in individuals who are poor metabolizers for CYP2D6. So this has been important. So you can imagine if somebody has their 2D6 knocked out, either due to a mutation, or this is why drug-drug interactions are also very important. Many drug-drug interactions are mediated through the same mechanism where they're inhibitors of 2D6. So whether you knock the 2D6 out by a drug-drug interaction or through a mutation, you're going to get reduced. We're going to get reduced desmethyl or endoxifen. So here's an example of the effect on pharmacokinetics. So this would be an individual that has 2 fully functional alleles. A 2D6 has very normal endoxifen concentrations. An individual who has 1 functional allele has reduced endoxifen. Well, that would be a bad thing to have reduced endoxifen. And an individual who has no functional 2D6 alleles has very, very little of endoxifen that is formed then from a pharmacokinetic perspective. This has been looked at now in terms of, certainly it's been known that there's been this pharmacokinetic interaction with the mutations for years, and there's been many, many studies now that has looked at overall survival and recurrence in breast cancer. It's very controversial, I'll tell you that right now, that whether or not that this should be actually an intervention. The guideline group did feel that there was sufficient evidence, though, to produce a guideline for tamoxifen. And what they concluded, and this is they have one that received a strong recommendation. All their guidelines, all their recommendations are rated. They gave a strong recommendation to for individuals who are CYP2D6 poor metabolizers. So that would mean that individuals who are poor metabolizers, they would have this 2D6 primarily completely blocked out, get very poor endoxifen formation. Those women should, if they have, in the case of breast cancer, should receive an alternative therapy, so like an aromatase inhibitor. Now this affects the frequency of 2D6 poor metabolizers is only about 2%. So this does not affect that many women. But you can imagine if you're that woman that you're a poor metabolizer for 2D6, this may not be what you would choose for your therapy. So 2D6 is also highly also metabolizes many of the opioids, and you can see how this would have a role in cancer too. But as you all know, codeine is a prodrug, just like tamoxifen is, and codeine is transformed to morphine. Morphine is a very, very good analgesic. So codeine, if you're a 2D6 poor metabolizer, you're not going to get morphine formed and you're not going to get the analgesic effect. Now, it's not only codeine that this relates to. Certainly the drug tramadol, which is another analgesic, also is biotransformed by 2D6. So individuals who have or are poor metabolizers for 2D6 don't get a lot of effectiveness from tramadol. There's emerging evidence now for hydrocodone and oxycodone. This is an active area investigation right now that perhaps could be genetic determinants. This has been, oxycodone and hydrocodone have not made it into guidelines yet, but they probably will at some point as the evidence continues to emerge. But codeine and tramadol, it's pretty clear that those people who have who are CYP2D6 poor metabolizers do not have a good analgesic response from those drugs. The other drug that's it's used commonly in cancer is the 5-HT3 antagonist, or Zofran. So it's used as an antiemetic to prevent nausea and vomiting with chemotherapy. And they've also graded, and they've also evaluated the literature for this and graded this. This has also received a recommendation. And because Zofran or Dansitron is metabolized by 2D6, individuals who have altered metabolism, especially if an individual is an ultra-rapid metabolizer, they're going to clear their 5-HT3 antagonists very, very quickly, and they're not going to get good antiemetic response from it. So they're recommending that those patients who are ultra-rapid metabolizers receive a drug like Kytril, because it's not dependent on 2D6 for metabolism. So moving on to the other CYP I want to talk about, 2C19. There's a number of 2C19 substrates that are affected by these mutations. Voriconazole, that is used in pediatric bone marrow transplant, so I'm going to talk about that one. Of course, the SSRIs, these are antidepressants that are often used in patients with cancer. Patients with cancer get depression too, and clopidogrel, which is often used in hematology. And there's a number of different phenotypes that are created from these mutations, all the way from poor metabolism to ultra-rapid metabolism. And I want to point out one more time that there's pretty extreme differences in minor allele frequencies and then therefore phenotype frequencies for the 2C19 substrate. So we can see that Caucasians and East Asians are very different in terms of the metabolism phenotypes that are created due to the mutations that they commonly carry. So just a little bit about what we know about voriconazole pharmacokinetics, and as it relates to CYP2C19 variants, we know that there is a profound effect of the CYP2C19 variants on pharmacokinetics of voriconazole. This is a drug that's used as an antifungal agent, so this would be for for cancer patients or other patients who might develop a fungal infection. They would require this for treatment. And if we look at individuals who carry 2 mutations within CYP2D6 or 2C19, they have much higher voriconazole levels in the blood. That's relative to individuals who are ultra-rapid metabolizers, and they have very low concentrations of voriconazole in the blood. So those with the very low concentrations, you expect that they're not going to respond as well to the therapy. And this has been highly associated with these mutations. The other thing that I think was really important that was recently described was that this was a group that had intervened, and so for depending on the genotype or the genetic mutations that they carried, these children carried, they gave them different doses of drugs. And what they found that time to therapeutic was dramatically better. So it was reduced. So you could get these children to a therapeutic concentration much faster if you use genotype-directed dosing. So that was some important evidence. However, that was just one paper where they have done that so far. So there are CPIC guidelines now, and they've also received a moderate level of evidence or a moderate grading. And they now they don't recommend that the dose is changed for voriconazole based on genomic mutations. They recommend that you just don't use it. So you use another drug other than voriconazole to avoid all these metabolism problems that we have with these mutations. Okay, the next drug I want to talk briefly about is 6-mercaptopurine. So this is sort of like the father of pharmacogenomics. These genetic associations have been known for decades now. This was really the first drug that became part of clinical practice of pharmacogenomic testing. This was pioneered by groups at St. Jude and at Mayo Clinic. They're the ones that did a lot of the discovery on these mutations. I'm not going to go over this complicated metabolic pathway, but there's 2 things that are really important. One, TPMT is one of the major enzymes that metabolizes 6-mercaptopurine, and it's really important that 6-mercaptopurine is converted to an inactive product through methylation. So anything that would knock out TPMT either through a mutation or other, that you're going to have greatly increased cytotoxicity. So these children are going to be at greater risk to develop toxicity if something goes wrong with this particular enzyme. Now, this has long been known that these mutations are much more common in European children of European and African ancestry. So there hasn't ever been very good markers for children of Asian and Hispanic ancestry. And that's where the new T15, this is the new mutation now that was discovered and now has made it into the guidelines. So now we know that this new T15 mutation, if that has mutations that create a loss of function, that becomes the risk allele for intolerance for children of Hispanic and Asian ancestry. So now both of these we know are incredibly important. There is a guideline, a CPIC guideline for TPMT and NUD-T. These are all extremely strong level of evidence. So many places are doing that, are doing genetic testing in their children before they give them 6-mercaptopurine for acute leukemia therapy. And essentially, its recommendation is that they get dose reduced. So if they have 1 mutation, they get a 30 to 70% of the target dose. If they have 2 mutations, if they're homozygous, then they go to 3 times weekly dosing of the chemotherapy. This is the new guideline, the NUD-T15 mutation, and the same thing is true. That in children who have 1 mutation, they get 30 to 80% dose reduction. In children who are homozygotes, they get dramatically decreased dose, 10 milligrams per meter squared per day. So, and then the final pathway I'm going to talk about, and this is because this also has a considerable evidence, is DPD. and this is involved in 5-FU metabolism. And this is an extremely simplified metabolism pathway for 5-FU, but we know that 5-FU that the majority of 5-FU is metabolized into inactive products. More than 95% goes into an inactivation, towards an inactivation pathway. About 1 to 3% of the pathway then will goes to metabolites that are responsible for the cytotoxicity. So anything that would reduce the activity of DPD is going to dramatically change the balance of this metabolic pathway and push things into the cytotoxicity pathway. So you can imagine then that it doesn't take much of a change in the DPD activity to get a big increase in the cytotoxicity, and that's the basis now for the guidelines and the rationale for dose reduction. I don't have the CPIC guidelines here, but essentially, just like in our case, the individuals who carry these mutations, they need a dramatically lower dose to tolerate therapy. But what I want to show you here, and I know this is a really busy slide, and you can't really see it that well, but one of the things that the Pharmacogenomics Implementation Consortium has done to help push us into using these guidelines into practice, is they've written clinical decision support alerts that can go into the electronic medical record. So they've created a lot of language for easy implementation into clinical practice. For example, it says that if a person is an intermediate metabolizer, this is what your electronic medical record then would alert the physician who would be ordering the drug. So if they had a mutation on the panel and they ordered the drug, then this little alert would show up and it would direct the physician as to what might be the recommendation. So these alerts are being built now, and these are freely available now for clinical implementation. And they're continuing to build these alerts for all the different pharmacogenomic drug-gene pairs. This was a paper that was just published this year, and I thought this was pretty interesting. This group had gone back and looked at all the published pharmacogenomic studies. And they went way back in time, clear back to the times when 6-mercaptopurine was first FDA approved. So that's like several decades ago. They reviewed all the pharmacogenomic literature. They then graded the evidence, in their opinion, had graded the evidence. And the lines that are in orange are the ones that they felt have sufficient evidence for clinical implementation. So 6-mercaptopurine, certainly they felt that there was sufficient evidence for that one. Vincristine, which, by the way, there isn't a guideline in the United States for vincristine. 5-FU, the amount of data for 5-FU pharmacogenomics is immense. They felt that was very strong data. Doxorubicin, tamoxifen, cisplatin, and irinotecan. So, in their opinions, these were the drugs that had the best and the most robust pharmacogenomic information. I work with a group in Canada, and they use the genomic markers for vincristine, doxorubicin, and cisplatin. So they have gone down kind of a different pathway, and they have implemented different drug gene pairs, and they're using genetic variants for drugs that we don't. That there's really not on our radar screen, and as far as I know, that there's no CPIC guidelines that are being proposed for those. So it really does depend. So there's a number of centers in the United States that have implemented preemptive pharmacogenomic testing. So Vanderbilt University has been one of the leaders in pharmacogenomic testing. They have a preemptive panel that they do in their patients. St. Jude Children's Hospital, of course, pharmacogenetically tests every child that is admitted. University of Florida has been really integral in moving forward, especially moving the needle on cardiovascular pharmacogenomics. University of Illinois, Mayo Clinic, of course, Mayo Clinic has done a lot of pharmacogenomic work. for now probably over 20 years. University of Maryland has led in pharmacogenomics, and Mount Sinai Medical Center has also. But what's interesting about all these different centers that have implemented preemptive pharmacogenomics is that they've all chosen different gene panels. So they really look at their institution, what kind of drugs are they using, what kind of procedures do they perform, and they've implemented different gene panels. And they've all used a different testing approach. Their laboratories are using all different testing approaches for those PGX markers. So there are a lot of pharmacogenomic testing companies now that have popped up over the last 5 years. Probably has been the biggest increase in commercial pharmacogenomic testing companies. There's not one that's necessarily better than the other. All really depends on what kind of pharmacogenomic marker that is of interest. 1-ohm happens to be one that's used quite commonly in this area. 1-ohm is a pharmacogenomic testing company that's up in northeast Minneapolis. They have a nice panel, so I understand why people use them. But there's a lot of different testing companies. There's also been big differences in terms of what these pharmacogenomic testing companies put on their panel. Some of them will have what they call a comprehensive panel, so they have lots and lots of different genes and lots of mutations. Others have chosen a business model where they might create an oncology panel, or maybe a pain panel, or a psychiatry panel, or a cardiovascular panel. So a lot of different business models right now in the pharmacogenomic company. They're trying to figure out what's the best, how they can probably make the most money. What kind of panel are people willing to spend their money on? And all of them have different costs. So I've been thinking a lot about what would be a reasonable pharmacogenomic panel that we could do here at the University of Minnesota. What would be something that we could implement here that I think would have relevance? Well, clearly, we'd want to have the CYP2D6 genetic variants on any type of panel. Those are important towards the opioids, the SSRIs, the tricyclates. You could add tamoxifen on there. CYP2C9, warfarin, is still commonly used, even though it's a really old-fashioned drug. We still use a lot of warfarin. CYP2C19 would be very relevant to the 5-HT3 antagonists, help us pick antiemetic therapy. The voriconazole, the proton pump inhibitors, there's going to be a guideline that will come out this next year on the proton pump inhibitors. And that evidence is looking really strong, too. So, of all the 4 or 5 different PPIs that are over the counter, which one might be the best to choose? The SSRIs, tricyclic antidepressants, and clopidogrel. So all of those are things that we would use. The DPYD, 5-FU, clearly, and capecitabine, both of those, this would be very useful in helping guide our therapy for DPYD. The HLA-B has a number of drugs that are used in oncology, like allopurinol and carbamazepine, so that would be useful in that. The NUD-T and the TPMT for 6-mercaptopurine. And then there is now a guideline for the RYR1 and the CAN-CA1S genes, and those are genes that have mutations that predict malignant hyperthermia in surgery. And of course, a lot of cancer patients go on to get surgery, so it would be good to have some sort of marker if they're at risk to develop malignant hyperthermia. And then irinotecan, I put a question mark next to that one because the evidence for that is a little bit weaker, and we don't really use high doses of irinotecan. And then, of course, the VCORC1, which is the other mutation that is important towards warfarin. So this would seem, and this list just keeps growing and growing of all the different genomic markers that are relevant in oncology. So, kind of changing gears just a little bit, I just want to mention that we're going to start a pharmacogenomic study in the Mille Lacs Band of the Ojibwe, which is up in Onamia, Minnesota. This study, it's at the CPRC and IRB now, but we hope to be starting that within the next few months. But it's important that we do this because we know that for many underrepresented minority communities, they have different responses to cancer drugs. Sometimes they don't respond as well. Maybe their outcomes aren't the same as what the typical Caucasian population might be. And this could be related that they have genetic mutations that maybe put them at lower risk to respond to some of the cancer drugs. So we're going to do a pharmacogenomic study. We're not going to be looking at disease risk mutations, but we're going to be looking at pharmacogenomic markers. So we're first going to look at the known pharmacogenomic variants. We're then going to do next-gen sequencing to see if we can identify some new pharmacogenomic markers that might be specific to a Native American community. And then we're also going to do return of actionable results. So we're actually going to give the patients back their pharmacogenomic results and explain to them what it means. So in conclusion, there's a number of challenges to integration of pharmacogenomic testing into practice. One, we need preemptive pharmacogenomic testing. It's not always done on a preemptive basis. Preemptive means that you do it before you need the information. What's happening right now in many places is the patient develops a side effect or they have an unusual response, and then the pharmacogenomic testing is done then in retrospect to try to explain why the side effect occurred or the non-response occurred. But to make preemptive testing, we need to have systems by which we can incorporate the information into the electronic medical record, which is not simple. To incorporate pharmacogenomic, any kind of genomic information into the electronic medical record so it's available at time of prescribing. We do have lagging evidence of clinical utility or cost-effective analysis. And of course, with any scientific innovation, cost-effective and clinical utility information always comes later. So we're really now in the phase of this new science where we're starting to get to the cost-effectiveness and the clinical utility information. There is, I feel, an imposition of higher than usual requirements for implementation and practice for pharmacogenomics. And then compared, I have heard people say that we need to do a randomized controlled trial for every pharmacogenomic marker. But that's not reasonable. There's 20,000 genes and there's thousands of mutations in all these genes. We cannot possibly do this many randomized controlled trials. So we have to think of other trial designs like retrospective analysis to prove the value or disprove the value of these markers. And then when compared with testing for Mendelian diseases, pharmacogenomic testing for certain indications has lower positive predictive values. So for example, for Huntington's disease, if you have the gene, the individual will develop Huntington's disease. Individuals who have these pharmacogenomic mutations are at higher risk. It doesn't mean it absolutely will happen. It just means that they're at much greater risk. So the positive predictive value is lower than what it is for some of these very strong inherited mutations like Huntington's disease. We also have a big problem with fragmentation of the U.S. healthcare system. So if Mayo does pharmacogenomic testing in a patient there, and then the patient comes and gets care here, the chances of us getting that pharmacogenomic information and being able to use it is very unlikely. We do not have a good way to share this kind of information across healthcare systems. So guess what happens? The patient gets retested. So it does lead to a much more expensive type of healthcare where we don't have a good system where we can share. We also lack expertise to interpret and apply the results of pharmacogenomic testing. In the College of Pharmacy, our students have pharmacogenomics has been a required part of the curriculum, I think now for 6 years. But any of the pharmacists that were trained before that may not have had any pharmacogenomic training. It's similar for medicine and for all disciplines where this is a new science. So we have the older workforce that doesn't perhaps know this. We also provide a elective in pharmacogenomics for students who want to learn more about this. They can enroll in our elective. That's new. We've only done that for maybe 7 or 8 years. We have lack of policies, reimbursement, and integration of new technologies into practice. It's just hard to create some of these tools that can be incorporated into electronic medical record, like clinical decision support systems. It's not trivial to develop those. So pharmacogenomics of future. So it's not a matter of if, but a matter of when, in my opinion. Preemptive, there's soon going to be a day where preemptive genetic panels are going to be conducted early in the individual's life. Everybody's going to show up to their doctor's office with some sort of genetic panel completed. I think this is going to be true for disease genetics, too. It's not just pharmacogenomics, but it's going to be all genetics, that they're just going to show up with a panel. Next-gen sequencing is going to be the standard platform, so we'll just look at everything. And personalized treatment and personalized dosing is going to be standard of care. At least that's my prediction. So this is how some people, and I happen to agree with this, is why I use this slide, that this is how they think of us when we use drug therapy. We have a lot of pills within that gun, and we shoot at these cells, but fortunately these people are blindfolded. So they're just hoping that the drug hits the cell and we achieve our response. But we don't. We know that. We know that not all people respond to medications. This is why. So I'm going to stop at that, and I'm happy to take questions.
That was a very interesting talk. I learned a lot. Thank you very much. So I have a general question. How often do you see significant between patient drug metabolism difference, but then there is no SNP to be associated with that? And then in those cases, do you recommend looking at combination of mutation in different genes or even non-genes in the non-coding region?
Yeah, well, between patient differences is fairly extreme with pharmacokinetics. We can see big differences. Now, for drugs that have a very big therapeutic index, probably doesn't matter. But for cancer drugs which have a narrow therapeutic index, this can be pretty important. But it's not all about mutations. There's certainly pharmacokinetic differences can be explained by demographic information, their age, their gender, maybe their renal function. There's all kinds of different. So what we've been doing, and some of my work has been, I work with the cytochrome P453A4 and 5 enzymes, and what we've been doing is creating models where we can incorporate all these determinants. So I think it's really we have found that our models are much more powerful if we take genomic information and incorporate other things like age and gender and drug-drug interactions we know are very powerful. Now what we're doing, we're going to be incorporating microbiome information. So we're going to incorporate our genomic information with all the patient-specific factors with microbiome information. So, it's a lot of things that matter. So we're naive to think that we can predict everything with a genetic. We're not.
Got a question over there.
Yeah, Ling.
Pam, really very interesting and very important work you are doing. So I have a question you have actually touched a little bit, because earlier in your slide, almost all the CYP, the cytochrome P450 enzyme appear to be indeed you had during aging they all going up. I saw the arrows, almost every one of them. So you're looking at the germline. What the germline mutations? What about epigenetics? So the people have looked into this because during aging, that has to be something else.
Yeah, the epigenomics is also going to be really important, and then Talia's here, so she could probably speak to that even better. But I think that that's just an emerging genomic too. I should have added epigenomics to my list of things that potentially has an effect on drug therapy. So it is important, Ling, but the evidence for that, they're not the evidence yet for implementation into clinical practice. But I think at some point there will be for certain drugs, absolutely. Yeah, Natalia wants to say something.
Very nice seminar. I would predict that epigenetics will also be very important. We just don't have the information yet. for a lot of the studies. My question was a more practical one. So you mentioned that in the future we'll all use next generation sequencing to uncover those polymorphisms. But what methodologies are used right now in those panels? Are they PCR based?
Yeah, lots of different methodologies. Many of them are PCR based. There are some groups that are doing next-gen sequencing now, or they're transitioning to next-gen sequencing. Now, not everything you can get on a next-gen sequencing. There's always going to be needs for alternative platforms, like to get duplications or deletions. There's always going to be other platforms. But most of them have been PCR, but they're moving on to bigger and better platforms now, many of these companies. They're trying to figure out what is the cheapest, the most cost-effective method that they can get good quality calls, because there's a lot of pressure to keep the cost of the panels down.
Yeah, it was a nice talk. So I'm wondering about, so we have a lot of new reporting showing that the gut microbiome does a lot of chemical transformations on drugs. How do you, or where do you see this field going with regards to that? Like, do we have any candidate genes with regards to the gut microbiome, or is that still naive in the field?
No. So we have 2 projects that are about ready to launch, and we're going to look at the gut microbiome and its effect on mycophenolate. So mycophenolate is an immune suppressant that we use in bone marrow transplant. And what we hypothesize is that the gut microbiome is very influential in interhepatic recirculation. So we need certain enzymes that are made by the gut microbiome to promote the intrahepatic recirculation. But in some patients, those microbes might be eliminated due to antibiotics or maybe even the anti-cancer therapy that they receive, that those bugs will be gone. So then they don't create enzyme, we don't get intrahepatic recirculation, and we have a dramatically decreased concentration of those drugs in the blood. So I'm biased because we're doing this work, but I think that the microbiome is going to be really important. So I think for some drugs it's going to be genetic determinants. Other drugs is going to be genetics plus microbiome. And for some drugs, genetics don't matter at all. Maybe it's just going to be microbiome or other factors. So, I mean, it's going to take a long time to sort out the relevant characteristics for each different compound.
So, very interesting talk. I guess when I look at this field, it kind of strikes me that the panel sizes aren't increasing in the way that they are in, say, somatic cancer events or in some of the other genome-wide associations where they're getting convincing associations. How large of a study would you need to go back and, at a genome level, a whole genome level, see these associations that are well known, like the SIPs? How large of a study would have to be to have enough power to convincingly see these associations relative to the sort of spurious associations you get when you look at hundreds of thousands or hundreds of millions of variants?
Well, I think for the drugs, we're fortunate because we often know a lot about their characteristics. We know how they're absorbed. We know how they're distributed. We know how they're metabolized. And we know how they're excreted. And we often know a lot about the mechanism of action. So in that case, I don't think we really need to be doing these really large panels. We can take more candidate gene approaches because you're right that if we were to take a whole genome or even a GWAS approach to some of these, we have to have thousands of patients to do this. We've been conducting GWAS, and we've been putting GWAS across pharmacokinetic information, and we have needed thousands of patients to show relevance to some of the mutations that have a low minor allele frequency. We know they're really important, but it took thousands of patients for us to demonstrate that. So, I can't give you an answer how many it's going to take, but it would take sizable. That's why most of pharmacogenomics is going down the candidate gene approach, where we take what we know about these drugs and we use our knowledge instead of trying to do whole genome wide. So I don't know if we could ever get to that, get to, satisfaction of satisfy the p-value.
Pam,
Yeah.
what do you think the reason is we maintain this heterogeneity in this set of genes? Is there an evolutionary advantage? And do primates maintain this, who mostly don't take drugs, maintain the heterogeneity in the same set of genes? Why do we do this?
Why is there heterogeneity? It's
Right.
probably evolution. I don't
For
know
what?
if I can even
For what?
answer. From what?
Yeah, what's in there's your substrates, and why do we need
Well, for example, in some of the African ancestries, there's some evidence that maybe it's some of the diet. Maybe there's some evidence for vitamin D. That maybe because of the sunlight, that the intense sunlight in some regions of the world, that they developed these mutations many tens of thousands of years ago, and those have been conserved. But now we don't need those anymore, right? So they just remain. So I don't think I can answer that well, Doug, because I don't know if anybody really understands it, why we have all of these, but we do.
Nice talk, Pam. I had a question with regard to clinical trials. How do you see the emerging of how doing this, how it's going to affect clinical trial development for new drugs, where you don't necessarily know, exactly what's going to happen in a phase 3 or phase 4, right? You finally pick up something. Is there a way, you see that a really intentional way of using this information, how to do this to really make clinical trials a lot better and more efficient, particularly in the early days, so that you can actually pick up maybe a phase 2 actually some of the potential problems, particularly I'm saying with new drugs, drugs where we don't have any, not 5-FU, something where
you don't know much about the ADME properties of the compound. It's going to be hard, right, if you don't know anything. But one of the projects that we've been working on, using CRISPR-Cas9 to insert mutations, certain single mutations into hepatic cells. And then you could take those single mutations, you can study one at a time, understand which of those mutations then in an in vitro drug metabolism system has an effect, and then perhaps use those in the clinical trial. So take that new compound, do your preliminary studies, and determine which of those mutations might be relevant. Then you could study a very small set of mutations, because you can't. You're right, Rick. We can't be studying a panel in a phase 1 where there's only 30 patients. We're just never going to pick up enough mutations to have any type of power. So we have to think of more laboratory-based technologies by which to identify which of those mutations are important, and then intentionally study those in the phase 1, phase 2, phase 3. Because all of this pharmacogenomic information, almost every one of the big, good papers has been from retrospective analysis, where it's done years after the drug has been developed, and it's been in clinical practice for a long time. So, it's kind of done in reverse, as it should, and it shouldn't be. But